In today's video, we're going to go into a second episode discussing Dr. Bosch's theory of how the pandemic, according to his theory, might become more pathogenic, something that I want to argue against. And I'm going to explore a new evidence that unfortunately is indeed supporting his theory. So let's get started. My name is Dr. Raszek of neurogenomics. I'm going to tell you a little bit about his theory. I'm going to tell you how you're supposed to protect yourself in case he is right and he believes that it is predominantly the mRNA vaccinated individuals that will be afflicted, and I'll explain that why he believes that. And again, this is just a hypothesis, so keep that in mind. And don't worry, if you're mRNA vaccinated, I got you covered. I want to argue this theory. I want to argue basically with him. And I'm going to then talk about this latest evidence that, in fact, he provided. I got this paper. He gave it to a bunch of us during one of his immunology courses. And it's really fascinating paper. We're going to basically talk about amazing evolution of the virus that is absolutely mind-blowing. I did one video in the past showing how virus can do amazing tricks in order to evade our immunity, and this one blows it out of the water. It's just next level. It's very, very fascinating. Absolutely amazing molecular engineering, mind-blowing. So and this will also be very picture heavy video, and it will be long, so bear with me. There's no way around it. It's a very complicated topic, but really fascinating story. And then I'm also going to introduce to you some of the technology that is being used to show how this data is obtained. It's also really fascinating. And okay, let's get going. Basically, the way the paper starts, I thought this was really interesting. They talk about that, how the variants are being selected. Remember, we constantly have this wave after wave after wave of new variants, right? Like, what is going on? Like, we've never seen that before, to be hit by this many variants, one after the other. Seasons make no difference at all, right? It just doesn't work like that normally, right? Or you typically get your influenza or colds in specific type of season of the year or time of the year. And right now, it doesn't matter. We are hit by these waves one after the other, irrespective of time of the year. So the authors mention that these variants are being selected specifically by being targeted or attacked by antibodies that are flowing in our body. And of course, these antibodies are produced because of the fact that we were either vaccinated or we were naturally infected, and hence we produce the antibodies. Or if you are vaccinated and then subsequently infected, that's called breakthrough infection. That's not supposed to happen. Technically, if you're vaccinated, you're supposed to be protected. Obviously, in this case, that didn't happen. In the case of COVID-19 vaccines, that didn't happen. And this is basically what breakthrough infections are all about. So hence why we call them breakthrough infections as opposed to natural infection. So all these antibodies, they place immunological pressure on the virus, and basically virus is always mutating because there's so many of it. When the virus is being produced inside our cells, literally thousands and thousands and thousands of copies can be produced. So as a consequence, mutations can easily be introduced. So basically, there's a plethora of different mutation options that the virus can use to select a variant that will help the virus to evade the immune system. So the reason why I found this interesting is because go back a couple years and no one would have even dared to mention that. At the time, we were under extreme scientific censorship. of the kind like I've never witnessed in my life before, and nothing could be said with regards to vaccines that could place vaccines in any bad light, including obvious stuff, such as obviously antibodies would be placing would be placing evolutionary pressure on the virus, but at the time it was a taboo to mention this, and here we are a couple years later, and this is being now mentioned by these authors. Why this is interesting is because the authors are also mentioning, look, eventually the virus is going to run out of potential options of how it can mutate, because there's a couple of things that can happen. The virus can mutate and change its amino acids that make up its viral proteins. Of course, the protein of interest is the spike protein. So that's option number one. And of course, by changing amino acids are the building blocks of proteins. There's about 20 of them, and it's the genetic code of the virus that determines which amino acids are being linked together in a row out of those 20 options to eventually produce a protein. So this chain of amino acids, these amino acids start interacting with one another, and as a consequence, what they do is they produce a three-dimensional entity, basically a tiny molecular three-dimensional robots, because proteins are basically like tiny little robots. They're not alive, but they can respond to the environment and move. And as it move to a point where they can actually act like tiny little miniature machines. And this basically how it works. And you can change these amino acids. Mutations in the genetic code can determine whether what amino acid is being incorporated in a protein production at what place. So that's one way to do this, but there's another way to do this, and that's through sugar molecules. So what do I mean by that? Basically, a lot of molecules in our biology are decorated with sugar molecules, and proteins are often also decorated with sugar molecules. Sugar molecules, now when I say sugar, you can often probably think of sugar that I'm maybe like adding to my coffee or something like that. But sugar basically is a very wide family of chemicals. It's a very broad family of chemicals, and they can be attached to many molecules. They can be attached to proteins as well. They can be attached to the surface of our cells as well. And by being attached to these areas, basically what you end up doing is you altered the three-dimensional shape of the entity, because everything in a molecular world is governed by two things: the three-dimensional shape and its electrostatic influence. So everything has some electromagnetic influence. So everything has some charge to it, and those two things will ultimately influence how things behave biologically on a molecular level. Okay, so that's basically what sugar molecules can do. And there's these sugar molecules are attached to proteins at two different types. These are called N-glycosylation sites. Now, N stands in this case for nitrogen, because in N-glycosylation sites, the sugar molecule is attached to a nitrogen of a specific amino acid. The specific amino acids to which the sugar molecule is being attached is called asparagine, and the code for that amino acid is also N. So hence it's a perfect name, N-glycosylation. And there's another one called O-glycosylation. Now, N-glycosylation, let me maybe tell you a bit more. It needs a specific sequence in order for it to take place. And that sequence requires the N amino acids, the asparagine, followed by any amino acid except proline. Why not proline? Because proteins are really weird amino acids. They're very bulky, they're very clumsy, they're not very flexible, so they're not, they just don't have the same biological flexibility, and therefore they can interfere with how the sugar molecules can be attached to the sequence. And then the last amino acids in the sequence is either going to be serine or threonine, and NX, T or S is the sequence of amino acids needed in order for the sugar molecule to be attached to that asparagine, to the nitrogen atom of that asparagine amino acid, also labeled as N. Okay, so that's one type of glycosylation. So we're talking about, again, attachment of sugar molecules to this motif. And the other one is O-glycosylation. Why O? Is because in this case the sugar molecule is attached to oxygen of an alcohol group or hydroxyl group, OH. It's called hydroxyl or alcohol. So, you know, when you think of alcohol, say alcohol you consume, it's going to be full of those OH groups in that liquid, right? Hence the colloquial name alcohol. And it's that oxygen molecule to which the sugar molecule can be attached. And this can happen on both serines or threonines, and that's it. You don't need any motif, you don't need any arrangement of any other amino acids. As long as serine or threonine is available on the surface for the sugar molecule to be attached, this could happen. So you would think that O-glycosylation sites would be more common, but that's actually not true. It's the N-glycosylation sites that are more prevalent. And the reason why is because of the fact that N-glycosylation happens during protein production. So as the protein is being made, and amino acids are literally being linked together, very soon after a number of amino acids are linked together, they're going to start interacting with each other and start shaping a three-dimensional entity, assuming that specific three-dimensional shape. And what happens at the same time while that happens, that's when the N-glycosylation takes place as well. So N-glycosylation as a consequence is very important for helping proteins assume three-dimensional shape as well. So they're added very early on in the production of the protein, versus O-glycosylation happens after the protein has already been made. So it's not as critically influential for the production of the protein as, say, the N-glycosylation is, okay? But clearly there's some flexibility there. So there's these two types of amino acids to which sugar molecules can be added. Now, sugar molecules can be added to many other biological molecules. They can be, like, as I mentioned, they can be also added to the cell surface of our cells. And for example, the SARS-CoV-2 virus can latch on to our cell surface via such sugar molecules. And I always joke that if you actually had a magical, I don't know, let's say light that you could flash somewhere to expose sugar molecules everywhere you look, you would see that there's literally sugar everywhere, because it's so prevalent, and these sugar molecules can be arranged into complex entities that can be arranged into these like tree-like or bush-like branches, and as a consequence, of course, they add additional charge and additional three-dimensional shape to whatever they're attached to. And typically, why would virus proteins desire this is for a couple reasons. Number one, if you attach these sugar molecules to viral proteins, it can help virus escape immune detection, because suddenly now you change again the three-dimensional shape of the protein, and now you have this flexible sugar branch entity attached to the viral protein. And another reason is to reduce the capacity of our body, of our immune system, to mount antibodies against it. So that's called immunogenicity. We'll be talking about this in greater detail in just a moment. All right, and another final reason is that it can help proteins with conformational change. What does that mean, conformational change? That's basically describing that the protein can shapeshift. So these amino acids, they're all linked together. Sometimes they're stuck in a rigid place, but other fragments could be very flexible. They can move about, and as a consequence, proteins have the capacity to dramatically sometimes change their shape, okay? And we'll talk about that in a moment. So the authors in this case also mentioned, look, clearly so many mutations have already happened in the SARS-CoV-2 virus throughout the evolution of this pandemic, that eventually you run into a limit, so that you might not be able to mutate anymore without compromising a virus ability, for example, to interact with a desired protein. So then the virus can adopt this other trick, which is basically the ability to attach these sugar molecules, okay? And they wanted to investigate the specific variant of the virus called BA.2.86, which is the parent of JN1 variant, which not that long ago took over entire North America, basically. And the reason why they wanted to investigate this, because that particular variant had not one, but two new glycosylation sites. All right, so they showed evolutionary tree of the virus. But before I show you that one, let me show you one from CDC website, because I really like the way they graph it. So in that image, you see the starting variant, the one that started the pandemic, and then it evolved eventually into something like Delta, and everything prior to Delta is no longer even shown on that image, because it's just too many variants, and the focus of that family tree of variants is now on Omicron. So Omicron came out at the end of 2020, I believe, and took over the world, and we're still in the Omicron family of variants. So we're still in an Omicron world. You can see BA.2.86 right there, giving birth eventually to JN1, and now JN1 is giving birth to a number of its own babies, so producing new variants. So that are currently fighting in the world for their own dominance, and there's a number of these, and there's not one specific variant yet that has dominated like JN1 did. But that's not unusual. What's more unusual is one variant actually taking over the world. We've only seen that a couple times from my memory recollection. That was when the Delta variant took over. Actually, Delta variant took over almost globally, and just recently JN1 variant also took over. So not a typical way of what's happening. So typically you have a number of different variants in unison together. So so that's the CDC graph. Now let me show you their graph. So in their graph there, it's much more complicated. They show you, for example, they show the different coronaviruses segregated into different clades, and they segregate them by color. So we're gonna ignore the blue one. So let's focus now first on the green clade, and I think you can see there's a variant coronavirus there, something like HKU. I think it's one of those common cold viruses, which is why I wanted to bring it to your attention. Now let's take a look at the yellow clade. And you can see in there coronavirus is labeled WIV. Now that's interesting because that actually stands for Wuhan Institute of Virology. So you'd be thinking like, why are the coronaviruses named after Wuhan Institute of Virology? And that's because they were literally making them in that institute. So they literally were taking different coronaviruses. Wuhan had the largest collection of coronaviruses in the world, and they were literally creating new versions of these viruses. And they will label them WIV. And so you see 1 and 16, and NIH was supporting some of this type of research via this company called EcoHealth Alliance, and of course that stirred a lot of controversy later on once the pandemic started. All right, so then now let's take a look at the red clade, because this is basically where the SARS-CoV-2 virus is present, the one obviously that started the pandemic. And right there you will see the label in bright red the protagonist of our story, the B8286 variant, which is what the authors studied extensively in this single publication. This entire video today, which I gotta warn you will be a long one, it's just on one single publication. And so that's in bright red, you can't miss it. Very close to that, you might be able to see coronavirus that is labeled something like RAT-G13. Now, what is that? That basically, in essence, is at the moment the closest known relative to natural relative to SARS-CoV-2 of any coronaviruses out there. Now, that's also a little bit interesting story, because the sequence of RAT-G13 coronavirus was published by Wuhan Institute of Virology. At the same time, they released the sequence of SARS-CoV-2. They were one of the first groups in the world to publish the genetic sequence of the virus that started the pandemic. Obviously not a surprise, because that's where the pandemic started, in Wuhan, right? But what's interesting is that this RAT-G13, they published fragment of that coronavirus multiple years earlier, and I believe that virus was isolated from one of the victims of workers who were working in bat caves, and multiple of these people who were working in those bat caves got severely sick, some of them with mysterious pneumonia, and some of them died. And of course Wuhan eventually got their hands on that virus as well. So and then not too far from that, you see a virus labeled something like PCOV, I think GX. That's one of those pangolin viruses. Now, pangolin coronaviruses. Now, pangolin's coronaviruses, from memory, as far as I understand, have now shown to have some of the most closely related spike proteins that we know of to the SARS-CoV-2, okay? So if you wanted to get SARS-CoV-2, the closest way you could achieve that is to take the RAT-G13 virus, coronavirus, remove the spike protein of that virus, take the one of the pangolin spike proteins, put that in, and insert the furin cleavage site, and you would get very, very close to obtaining SARS-CoV-2 virus, which is one of the reasons why, because Wuhan Institute of Virology had their hands on all of that, why there is now these continuously persistent rumors that perhaps SARS-CoV-2 might have been engineered in a lab, while we don't have any clear-cut evidence for that. Now, obviously, in that clade, in red clade, now you can see they zoom in with a pink color, and that's basically within the pink color you see the evolution of the SARS-CoV-2 during this pandemic. And basically the way it works is that the closer the distance between those lines is, the more closely related the viruses are, and the larger the distance, the longer the lines are on that graph, the more distantly related these coronaviruses are, okay? All right, so now let's look at another of these figures, and And now let's take a look at the amino acid sequence so that we can actually take a look what happened with those glycosylation sites. So on another image, so amino acids, they obviously have, each one of them has names, and they also have symbols. And the symbols are usually single letter code symbols, and this is exactly what they're using in that figure. And let's focus again in that image. You can see the clades in the same color as in the graph that I was just in that evolutionary graph I was just showing you. So let's focus on BA286 again, because again, that's our variant of interest because it has those two new glycosylation sites. And again, remember, BA286 gave birth to JN1 variant. That's the variant that just not long ago was completely dominating North America, and it's still being identified in North America to a certain degree, okay? And then remember the motif for the N-glycosylation site is N, X, S, or T. And then so if you take a look at BA286, you can see in the square brackets where there are different identified glycosylation sites. So let's take a look at the first one is around residue 245. That's in N-terminal domain. Don't worry, I'll explain what that means in a moment. And basically, it's the mutation of an N that incorporated that new glycosylation site in that N-terminal domain. We're gonna move forward to the right, and then you can see at residue 345, that's where the glycosylation sequence is present again. And here, it's not the 345 that was mutated, it was the residue two amino acids down, residue 346, 354, 356. And that one was mutated, and that's how you introduce that second glycosylation site. And that's the one that is key to our story. That's the one that did incredible magical things to these variants. All right, so then there's one more thing, and then there's also loss of glycosylation sites. So you can see that all of these coronaviruses that have another glycosylation site starting at residue 370, except the SARS-CoV-2. And it's believed that for SARS-CoV-2, what happened is there was a mutation that resulted in loss of that glycosylation site, and that loss of that glycosylation site helped the SARS-CoV-2 virus attain open conformation or up conformation. So what does that mean? So as I mentioned, proteins have certain flexibility that can change their shape, right? Or conformation. So what happens is spike protein looks like a mushroom. The viral spike protein kind of looks like a mushroom, right? So, and basically, when the virus interacts with our cells, first it needs to attach to itself to our cells. It can achieve that usually via binding to some sugar molecules on the cell surface. Then it needs to be clipped in a couple places, and once it's clipped, what happens there, basically it allows spike protein to be separated into two separate units, two separate subunits. It's called S1 and S2. How does that happen? Then basically the spike protein has to interact with some receptor protein. The most common one we know of is the ACE2 receptor. And once ACE2 receptor binds the spike protein that's been clipped, it can rip off the head. So that's the S1 subunit. Literally the head of that mushroom part of the spike protein will be ripped off, and it exposes the S2 subunit or domain of the spike protein. And that's where the magic happens, because S2 domain of the spike protein has these extendable arms. Remember, I was telling you proteins are like tiny molecular robots. They truly are like little robots. And the S2 subunit can extend its arms from its viral surface and grab with those arms cell membrane of a cell that the virus wants to invade, and literally bring the virus close to the cell, and then the virus can fuse with the cell. That's called fusogenicity. We'll be talking about that in some detail as well. All right, so basically, in order for all of this to happen, ideally for that head, S1 subunit, the head of the spike protein, to be ripped off, you want the receptor binding domain, or the area of the spike protein that interacts with the receptor on our cells, the ACE2 receptor. It needs to be in an up conformation. So that receptor binding domain, it's flexible. It's like I always say, imagine it's like a flower that can open up its petals or close its petals. So when it opens up its petals, it's in the up conformation. And when that happens, ACE2 receptor can grab it and rip off the head. Why does that happen? Is because when it's in an up conformation, it all becomes more loose, and the head is much more easier capable of coming off, okay? And if it's in down conformation, it's all more tightly packed, and the head does not come off. And remember, of course, you need to clip the spike protein too. You need to cut this at couple sites. One cut is done by tempress protein, and the other one is done by furin protein, okay? So basically, the loss of this glycosylation site at 370 helped the spike protein attain the up conformation much more easy. So basically, it was able to shapeshift itself in that position of up, and we'll see that in diagrams in detail. So that's a bit of a background for you, just so we can continue with this complicated story. And I haven't even started yet, really. I told you forewarning that this is going to be complicated and long, but it is really mind-blowing story. So because of that, what the authors really wanted to see is they wanted to actually determine what is happening here, and they decided to get a structure image of this BA2A6 variant. And now I'm going to introduce to you this wicked technology that they're doing. I've introduced this in one of my videos in the past, and basically this technology is phenomenal. Basically what they can do is they can take biological material, so in this case tons and tons and tons of spike proteins on the surface of some membrane, and they can freeze it, and then they shoot it with electrons, and the electrons will literally bounce off of everything. And depending on how far the electron travels in order to bounce off, it leaves a certain shadow, and the shadow tells you three-dimensional pattern of what it just bounced off of. So you can get a detailed three-dimensional pattern. So I'll show you an example of this. So they did this for a number of variants, but let's focus on BA286. And you can see these black and white images. There's these kind of weird blobs, right? That's actually what you're looking at is an image of a spike protein. It's basically as close as you can get of taking a photograph of a spike protein, except they take thousands upon thousands upon thousands and thousands of these. And I'm not exaggerating. This is the reason why I said it like that. And then because they took thousands of these, the computer can take all of these different thousand images, combine it together, and measure all these tiny little differences in shadows to determine exactly what the spike protein looks like, down to atomic level. So they can determine exactly what these spike protein looks on a molecular level. It's unbelievable. It's so cool. Now, it's not trivial work, but it's really, really amazing, because basically it allows you to study how the spike protein for any given variant behaves biologically. What kind of conformations does a spike protein attain? And when they did it for BA286, you can see then they took the computational images, and they were able to determine this image of a spike protein. And what you're seeing there is an image of a spike protein from a side. And what you're seeing there is that there was two conformations that the spike protein had. One was where the spike protein was an up conformation, so the receptor binding domain on the very top surface of the spike protein shifted up, and basically the part of that receptor binding domain just is sticking up, and about 21% of the spike protein was at least one of these receptor binding domains was in that up conformation, and then approximately 79% were all closed conformation. So all of the receptor domains were down. So not what you want to see typically for infection. And this is like makes you start wondering what the hell is going on. Why do I say that? Is because this is highly unusual, and the reason why is because typically throughout the entire pandemic, all the variants that have been infecting us, starting from the wild type that initiated the pandemic, and all the other different variants, wave after wave that we've seen, typically at least 50% of such spike proteins on these viruses would have been in the up conformation. Why? So precisely so that they can properly interact with the ACE2 receptor, and so that the head can be ripped off. And once the head is ripped off from the spike protein can then be used to engage in infusogenicity and basically infect. Virus can infect our cells. So 21 is now on a very low end. But that's not even the crazy part. They also did the JN1 variant, the one, again, remember, this is the one that just recently took over an entire North America. And crazy enough, and this I couldn't believe it when I first saw it, but this is really, really cool paper. 100% of JN1 variant was all in closed conformation. So what the hell is going on? How can you have a variant that became so dominant and yet it's in close conformation, meaning that it's not supposed to easily infect at all? So what is going on, right? So they also took a look at the same, and I'll give you an answer to that right away, but they also took a look at the image of this from the top. So now we're going to take a look at the spike protein from the top. So imagine if you were hovering over the virus, that's what the spike protein would look like. So when you're looking at the spike protein from the top, it looks like a triangle, and the very tips of the triangle, these little knobs at each corner of the triangle, those are the N-terminal domains. Why is there 3 of them? Because spike protein is actually made up of 3 identical proteins. So they're called monomers, and a spike protein itself, when you put 3 of them together, is called trimer, and more specifically homotrimer. Homo meaning they're all the same. So homotrimer, and then so that's your N-terminal domains, and you can see this blue entity sticking out. That blue entity is the sugar molecule attached to the N245 amino acids on the N-terminal domain. And then you can also see this red entity, and that's the glycosylation site at residue 354. And you can see that one sneaks in between N-terminal domain and receptor binding domain. Now, the middle part of this triangle, those are the receptor binding domains. They form like a circle. You can see there's like a gap in the middle, a little bit of like a gap in the middle. And there's 3 of them as well. And this is why you have 3 receptor binding domains. And basically that means spike protein could interact with up to 3 ACE2 receptors at the same time if all three of these receptor binding domains were to move into the up conformation. Now, I forgot to also tell you that when they showed us the image of the spike protein from the side of both the JN1 and which was 100% in closed conformation, and then the BA2A6 was 20% in the open conformation and 80% in the closed conformation. You can see these little tags sticking out there, right? Those are the different glycosylation sites on the spike protein themselves. So as you can see, spike proteins in general, they can have many of these sugar attachment points. Okay. Now looking further, what they also did is they overlaid the image of their BA286 with the image of XBB 1.5 variant. Now, XBB 1.5 variant, that's it was around a while back now. X means basically that two different variants recombine their genome together. So two different variants came together, and one variant gave portion of its genetic content, and the other variant gave portion of its genetic content, and they combined those two genetic contents together, and you had a new variant. So there you don't just can mutate specific amino acids. Sometimes you can literally exchange big chunks of genetic code as well. So that was designated with an X. And basically when they overlaid the spike protein on top of each other like that, what you can see is that the spike protein of BA286 becomes tighter, smaller from the top. It actually constricts. So, and it moved inwards by approximately 6 angstroms. And I'll tell you right away what that means. So basically it's more tightly packed. So what are angstroms? Angstroms is basically a unit of measure on an atomic scale. So whenever atoms are within few angstroms of each other, their electric charges can already start influencing one another, and this is basically exactly how proteins attain their three-dimensional shapes. Now, what is an angstrom? Angstrom is a 0.1 nanometers. Now, what is a nanometer? Okay, you're probably familiar with a millimeter unit of measure, right? Let's say my pinky fingernail is 10 millimeters, right? If you take 1 millimeter and divide it by 1,000, you're gonna get 1 micrometer. Take that 1 micrometer and divide it by another 1,000, then you get a 1 nanometer. And then angstrom is 1/10 of that, 0.1 nanometer, right? So that's basically where we get these units. So we're really talking about tiny molecular level, atomic level distances here. But basically what it meant is that the spike protein is more tightly bound, and they realized that it's this tight packaging that results in this completely closed conformation that they're observing with something like BA286 or JN1, and they have this amazing figure where they literally basically they start mapping how much do different receptor binding domains of the spike protein end up interacting with each other, and which they labeled as BSA, or I think it was buried surface areas, what that stands for. And basically the more surface area between the different receptor binding domains that is interacting with each other, the more likely it's going to be able to retain close conformation. And they map that. So they, for example, they show the wild type, the variant that started the pandemic, barely had any interactions between the receptor binding domains. Then the wild type mutated. One of the very first mutants is the one that they show on the very first variant in that graph. And that one had even less surface area interaction. This is why that mutant was quickly retained in a population, because it allowed it to obtain that up or open conformation even easier. But XBB1, as you can see, has very little surface area interaction, but BA286 has a lot of surface area of interacting between the different receptor binding domains, and as well as some of the animal viruses. And they produced this amazing graph where they actually map surface area and of interaction between the receptor binding domains and how likely they are to be in an open or closed conformation. And basically what they're saying is that if you have more than 400 angstroms squared interaction between the receptor binding domains, it means that the spike protein will be only in the closed conformation, and JN1 achieves that. So very cool figure. All right, so then they hypothesized, look, clearly what this means is that this glycosylation site must be somehow influencing the conformational state of the spike protein, and therefore it's likely to affect its infectivity as well. So they wanted to take a look at that, and what they did is they first what they did is they tested variety of different, they tested the variants for ability to infect, and indeed BA286 had a reduced infectivity. So clearly something is off here, right? Like, why are we having variants that are now in close conformation and are less infectious? So what's going on here, right? So the next thing what they wanted to take a look at is how well do these variants interact with ACE2 receptor? Does it affect its interaction with the ACE2 receptor? So what did they do there is they basically studied the binding of these different variants to ACE2 receptors. And here I was a little bit surprised, because what they found is that the introduction of this N354 glycosylation site did not do anything for the binding to ACE2, meaning that these variants such as JN1 or BA286 were able to bind to ACE2 receptors just as well as variants that didn't have this glycosylation site. So to summarize, is we having variants that are now more in closed state, can still bind very, very well to the ACE2 receptor. So all of these glycosylated N354 glycosylated variants, they were still binding to ACE2 super well, and they have reduced infection rates. So what is going on, right? Still head scratching. And then basically next what they did is they wanted to, of course, they went back. It's like, okay, let's get this structure. So they wanted to get a structure of BA286 bound to ACE2 receptor, and they produced this very beautiful figure, and they showed like, yeah, BA286 can bind to ACE2 receptor. So you can see this on the left-hand side, how BA286 variant can interact with not one, but even two ACE2 receptors. But it's the right-hand figure that is really, really cool. Basically, it's the best summary of the entire pandemic history of virus evolution, if you will, because it neatly presents to you of how the virus has been evolving throughout the entire pandemic. It's been almost 4 years now, right? And how it's been evolving in terms of its ability to bind to the ACE2 receptor. And as you can see, over time, it's been becoming more and more sensitive to bind the ACE2 receptor. Sometimes it goes up and down, but overall the trend is that basically the virus all this time has been evolving in order to bind the ACE2 receptor more and more efficiently. Basically, the higher the number, it means you need more protein, spike protein, in order to bind to all of the ACE2 receptors. And the lower the number, it means you need less spike protein in order to bind to all of the ACE2 receptors, which basically means like the binding is more efficient. So that's quite interesting. So obviously we're still having a bit of a confusion here, but in essence, this N-glycosylation site seems to be packaging these spike proteins in a tight conformation, locking them like a bolt. It locks the spike protein in a closed conformation. But they also knew that, look, as the virus has been evolving, spike protein has been evolving. Remember, each amino acid can carry certain electric charge, and the spike protein, as it's been evolving throughout all this time, has also been becoming more and more positively charged. And as a consequence, and I did a video on that in the past as well. And as a consequence, what the authors have mentioned is like, look, it's also been known that there's been more and more evidence showing that the spike proteins are capable of interacting with certain negatively charged molecules such as heparin sulfate that can be found on the cell surface of some of our cells. So I've also done a video on that long time ago as well. So they wondered, like, is there a correlation here? So they decided to look at this. So what they did is they decided to see how these variants infect the cells, but they started to add a little more of heparin sulfate to the mixture. And basically, while BA286 did not infect cells very well, it was much more weaker at infecting cells than other Omicron variants, much weaker than, say, XBB 1.5, as they were adding more and more heparin, it actually improved BA286 variant's infectivity. Not only did it improve, eventually, not only did it match infection rate of XBB1, eventually, as they kept adding heparin sulfate, it even surpassed the ability to infect. So suddenly, now you finally have solved the riddle. Somehow, this virus has amazingly evolved to be able to retain close conformation, tightly packed, and basically it means that this virus has evolved to now infect cells that would have heparin sulfate on them. So that means virus is now finding completely new ways of infecting our cells. Very, very fascinating. That's called tropism, how virus is changing its ways to infect us. This is referred to as tropism. So obviously they were quite interested in this. So obviously they were going to get a structure of that as well. So back to the cryo-electron microscopy we go. And here I'm going to show you some of their rat data again, because it's really fascinating. So they decided to get the crystal structure, three-dimensional structures, these from electron microscopy for the BA286 variant. They also did one for JN1, and they also did one for XBB 1.5, and they also did one for BA286 that was mutated so that that glycosylation site was gone, all right? And they added heparin sulfate, and they did this electron microscopy to see what they got, and they blasted it, and they obtained these structures. And one thing you can see is that clearly this showed the authors that what's happening is that addition of heparin sulfate also increases the likelihood of the spike protein of being back again in that up conformation. So super interesting. And you can see heparin sulfate in their three-dimensional structure. They colored it with the red, and you can see how amazing this technology is, that you can get such detail down to such atomic level, where in this crazy cloud of weird bumps and valleys and these weird features, basically what you're looking there is how the atoms are positioned in space, right down on the surface of the spike protein, right? And then heparin sulfate is shown there in red. And basically, heparin sulfate sneaks into this groove that is made between receptor binding domain and terminal domain. And they showed us now another nice complex structure from the top. And in this case, what we're seeing is now you're seeing the spike protein from the top, but colored based on charge. This is a very common way of how protein structures are presented in literature with charges. So red is negative charge, blue is positive charge, and white is neutral, like no charge. When there's no charge, there might be tiny amount charge, but basically negligible. But to me, basically the way I think almost everything has some level of charge on it, right? And you can see the center of the receptor binding domains is that right in the center of the spike protein is very, very, very blue, basically very highly positive charge. You can see that, and terminal domains, basically the corners of the triangle, are highly red, and is also heparin is there as well. Remember, heparin is highly negatively charged, so that's again shown in red, and the glycosylation site is shown in pink, and they also put an extra gold star for easier visibility. And you can see heparin sulfate sneaks in there. And they also did the same structure for the BA286 that was mutated, where there is no more glycosylation site. That N354 glycosylation site is now gone. And you can see the heparin basically binds in the same area. But there is actual small change. So remember, as I mentioned, that additional of heparin sulfate to the spike protein flips the spike protein into the up conformation. So remember how the BA286 was only 20% of it was in the up conformation? Well, with heparin sulfate, up to 60% of it was now up in an open conformation or up conformation. So more favorable for ripping out off that head, so that the fusogenicity, or basically being able to fuse the virus with the cell, is taking place. And for JN1, that's a big one. Remember, 0% of JN1 was in up conformation, or the shape of or open shape. And after addition of heparin sulfate, up to 40% of it is in that up or open conformation, right? So huge change. So basically, heparin sulfate acts like a key mechanism, where it's a key that goes into a lock and opens the spike protein. So imagine how this protein has mutated, that it introduced glycosylation site to keep the protein in a specifically closed conformation, and it now requires a specific molecular key in a form of heparin sulfate in order to open it, so that then it can finally become fusogenic. Isn't that crazy? And they studied it in more detail, and they obviously were able to get the detailed structure. And this is really fascinating. When you have the glycosylation site, that N354 glycosylation site, when you introduce that glycosylation site, it brings the receptor binding domain and an N-terminal domain a little closer. So the gap between those two domains is more narrow than what you would see without that glycosylation site, okay? But that also means is that when heparin sulfate sneaks into that groove, because there is that glycosylation, that sugar molecule in there, heparin sulfate has to adopt different position. And because it adopts that different position, what it does, it actually forces to push the receptor binding domain more away from the N-terminal domain by approximately 3 angstroms, and that makes the whole protein a little bit more open, more loosey-goosey, and now it's more capable of switching to the open or up conformation, because the receptor binding domains are no longer interacting with one another to the same degree when you snack that heparin sulfate. Now, if you take the BA286 that's mutated, so the glycosylation site is no longer present. You no longer have that sugar molecule in there. The groove is a little bit wider, and heparin sulfate can just enter in there in a slightly different position, and doesn't cause any movement, does not force any further distancing between the receptor binding domain and an N-terminal domain. So it does not act like this key. Versus if you have that sugar molecule, that glycosylation site at N354, it acts like a key, goes in there, and then it flips. And as a consequence, it opens up the protein. Absolutely amazing, and it just shows you how stunning the evolution of the virus can be in order to attain completely new methods of how it could be infecting us. Very cool. Absolutely mind-boggling in my opinion. I made a video in the past on how the virus is using sugar tricks, and this was related to, believe, one of the Delta variants, and this one is far more imaginative. This is so neat and so bizarre that this is one of the reasons why I believe there, like some people like to spread these rumors or conspiracy theories that these variants are being built on purpose and released to the public. And I'm like, and my idea is like, no, we're not that smart. Nature is way smarter than us, because we would have never thought of this incredible design. So and here's the magical explanation as to why evolution has evolved this. And look, this JN1, which is normally can be in 100% closed conformation, again, remember, took over North America not that long ago. All right, so then one more thing that I did, a couple more things that I wanted to tell you about. So basically, if in order for virus to infect, obviously that head has to be ripped off, right? So that means the protein has to be cleaved by furin. Now with furin, this is really interesting thing, because furin also has been mutating throughout the evolution as well. So in this one image that they showed, you can see how the sequence of the furin has been changing, and the bottom right corner of this image, you can see what this furin cleavage site would look like in the wild type variant, meaning the variant that actually started the pandemic. So you can see mostly it's blue. It literally sticks out like a little nip off away from the spike protein. You can see this little image of scissors. That's basically where the cleavage would take place. And you can see it's mostly blue, meaning it's mostly positively charged. And you can see a little bit of white there, and that one where it's white, that's that specific residue or amino acid has also been mutating throughout the evolution of the pandemic. So on the bottom left corner, you see one of those mutations that we've previously seen in alpha variant, as well as most of the Omicron variants. And then the top right corner, you see the mutation that we've observed previously in the Delta variant. And you can see that white component now is becoming blue, meaning the mutation that took place introduced a very positively charged amino acid. That's actually important because we know from the past that Delta was more infectious and was also more prone to be cleaved and also more fusogenic. And I'll talk about that just in a moment. Why is that of relevance? Is because that same mutation is also present in BA286, right? So that same mutation in that furin cleavage site is also present, and that was observed in Delta, and one of the more dangerous variants we've witnessed is also present in BA286. Now with Delta, I think the only reason why we haven't seen more deaths with Delta, which was more infectious and more fusogenic, is because Delta came about after mass vaccination started. So, but soon after, maybe a few months after, did it take over the world. And at the time, those who were vaccinated were still potentially fairly well protected with their amino acids. This is also where the theory of the pandemic of the non-vaccinated came about. The reason why is because imagine those who were not yet vaccinated, if they were infected with Delta as their first variant, it was even worse off than the wild type variant, the one that started the pandemic, which was also dangerous. So those who were infected and non-vaccinated with the wild type variant first, they already had the antibodies to be protected against Delta. But if those who were never infected yet and Delta was the first variant to hit them, well, that was a dangerous variant. For some of them, that might not have been good news at all, right? So that's basically that's where that entire story came about, until, of course, Omicron came about, which basically the immune escape variant, which then was so heavily mutated in comparison to the wild type variant that anyone was getting reinfected all over again as if they've never had any antibodies, right? And finally, that ridiculous story ended, that non-vaccinated were causing the pandemic. It was literally simply had to do with how rapidly the virus was mutating in comparison to how rapidly people were still being infected. By now, probably there's no one in this world who hasn't been infected, or almost no one, right? Because of how infectious the virus has become, as you have seen even in this paper. So Delta was a dangerous variant. And so the authors wanted to see, does this BA286 with this Delta mimicking mutation, I think it's position P or 681, and it's mutated from P to R, P being the proline to R, and R being arginine, one of those very highly positively charged amino acids. And indeed, when they looked at it, they were able to see that that BA286 splits into two halves, and S1 and S2, more readily than some of the other Omicron variants. Not as much as the wild type or Delta, but better than some other Omicron variants. But here's an interesting part. If they remove that glycosylation site from the BA286, it no longer split in those two halves as well. So clearly what that means is that these two mutations work in tandem together to help out the ability of this variant to infect us. So that's very interesting. And of course, remember, once you rip off the head, for infection to take place, you also need to have that fusogenic event. So basically meaning those, once you rip off the head, which is S1 subunit, and you are left with the S2 subunit, which is still attached to either the viral envelope or to the cell surface, then those arms of the spike protein will extend and they will grab the cell surface and pull it towards the virus. Or if the spike protein is on the cell surface, it will pull one cell towards another, and these cells will literally fuse together. That's called syncytia. When a number of these cells can fuse together, it's called syncytia, and they will literally have multiple nuclei. That's one of the hallmarks of early pathogenicity of this virus. And this is exactly what Dr. Bossche believes, is that that's what is now protecting us, is these certain antibodies that prevent this fusogenicity inside our lungs. And this is the only reason why right now, according to his theory, why Omicron variants are so much milder than what we've seen before. And I'll explain in a moment as to how he believes the virus will evolve to go around it. But so basically fusogenicity is key towards pathogenicity as well. And the more fusogenicity you would see inside our lungs, so the more lung cells can fuse with one another and create this weird blobs of cells fused together, the more pathogenic the virus becomes. Okay, so, and you can measure syncytia, and so they decided to do that as well. How do they do it? They basically take, oh, and by the way, I should mention before I go on there that vaccinal spike protein cannot do this. So vaccinal spike protein had introduced two proline mutations in specific locations. Remember I mentioned those proline amino acids are very awkward, bulky, and non-flexible amino acids. Well, the makers of the vaccines introduced these proline amino acids in specific places so that these arms of the spike protein could not longer extend. The head, the S1 subunit of the vaccinal spike protein could still be ripped off, so that could still come off, but the arms of the spike protein could no longer extend to grab an adjacent cell and cause fusion of cells. So vaccinal spike protein cannot do this, but viral spike protein could, okay? Anyway, going back to the measure of fusogenicity, the way you can do it is you can use cells that have spike protein on its surface, but the cell inside can have green fluorescent protein, meaning if you hit it with specific wavelength of light, you'll see green fluorescence. And when these cells fuse together, the green fluorescence can coalesce, and you can really measure how likely cells are fusing together. And they measure a variety of different variants that way, and they were able to show that the wild type and Delta was the most fusogenic protein there was, and we've known this already, even more so than the wild type. Omicrons, BA1 and BA2, which is the variants that started the entire Omicron family, they were not that fusogenic at all. But as you can see, BA286 is much more fusogenic than the earlier Omicron versions. So this is actually supporting Dr. Bosche's theory that these variants are, that the virus is mutating to eventually become more pathogenic, because you can see the fusogenicity is increasing, and this figure showed this very well. So the spike protein is being separated easier into different subcomponents, and it can now fuse even more easier, fuse cells even more easier. So again, hallmark of pathogenicity. There's exactly what Dr. Bossche is predicting. Now there are a couple more things, and I'm almost done. warned you that this is going to be long. So number one, what they also did is they basically noticed that addition of the specific glycosylation sites in the BA286 variant also prevented binding of certain neutralizing antibodies to the spike protein. Now that is to be expected, not a surprise. This is exactly why viruses have these glycosylation sites. But why do I mention? Because Dr. Bossche believes that exactly what will happen is that the virus will mutate and introduce new glycosylation site on its N-terminal domain, where at the moment he believes specific antibodies are binding to N-terminal domain of the spike protein. And as a consequence of that binding, these antibodies are preventing the virus to fuse with the cells in the lung. And this is why it's no longer as pathogenic. But as soon as this new glycosylation, and he believes this will be O glycosylation type, will takes place on an N-terminal domain and will prevent these antibodies from binding to the spike protein and therefore no longer be able to inhibit spike protein from participating in its fusogenic behavior, that's when we're going to have this increased pathogenicity. And at that point, he worries that this will become so deadly that the pandemic will become much, much more deadly. And why does he believe that non-vaccinated will be okay, provided that they're healthy, and mRNA vaccinated will be in trouble? Is because of the fact that when people were vaccinated with COVID vaccines, they produced a lot of antibodies, and this amount of antibodies that they produced prevented together in how they ended up interacting with the virus. It prevented proper training of our innate immunity, versus non-vaccinated individuals have their innate immunity trained because they've been exposed to small doses of the virus. They've never produced same level of antibodies. They had different immunological responses to the virus than the mRNA vaccinated individuals had as a response to the vaccine. And as a consequence, non-vaccinated individuals have intact and well-functioning innate immunity that will be protecting them, versus mRNA vaccinated individuals do not have this innate immunity to protect them. So once the virus mutates by introducing these new glycosylation sites and will increase pathogenicity, mRNA vaccinated, according to his theory, will no longer have either antibody protection or innate immunity protection, and he expects that there will be a lot more death. And this is why he proposes that you have to protect yourself specifically. And this is the most important message from Dr. Bossche. You have to specifically protect yourself with antivirals. If you truly worry about this, if you think this might be real, then get your hands on antivirals. What kind of antivirals? Well, the ones that before the pandemic was known to be as one of the most safest drugs in the world. One of the drugs that was on the top 10 list of the most important, top 10 World Health Organization list of most important medications known to humanity, and also the drug that has won Nobel Prize in medicine for its discovery. I assume you know which one I'm talking about, y'all, and or you can research your other versions of antivirals. That's definitely by far the most important message of Dr. Bossche in terms of how the mRNA vaccinated individuals supposed to be protecting themselves, if he is correct. Now, I don't believe he will be correct on this, and I want to argue him. Hopefully, we're going to have this as a live debate. I'm not ready for this because I'm still studying, preparing for debating him, because the guy is a genius. And look, I'm smart, but I'm not that smart. The guy is gonna wipe the floor with me, and I know this, but I really, at least if I'm going to go and argue him, and no one has yet dared to argue his theory, no one. So, and he deserves it. He deserves someone arguing him, so I better be well prepared. I'm studying like crazy for this, and I'm hoping we'll have a live debate on this eventually. So I got you covered, mRNA vaccinated individuals. I don't believe he will be correct. But basically, that's how he believes what might happen, all right? So, and the last thing they also studied, second last thing the authors also studied in this paper, they also showed that the BA286 variant also had reduced immunogenicity. What does that mean? It means that if you were to take this virus and inject it, let's say, to an animal that was never exposed to the virus ever before, and observe what kind of antibodies it's producing to different variants, it's basically, it means that the BA.2.86 results in lower number of antibodies produced against it than other variants. So this glycosylation site achieves three different things. Number one, it clearly attains new capacity of how the virus can now infect us. So this enhanced tropism. Number two, it's also it's escaping immune detection, so this immune evasion, because fewer antibodies can now recognize and bind to the spike protein. And number three, it has now it reduces the number of how many new antibodies can we produce against the spike protein as well. So it's a winner from multiple angles. Now, the very last thing that they did that I also want to mention, because I think this might help explain why maybe Dr. Bossche is not correct about his timing, is this was really interesting. The authors finally looked at basically history of how often viruses are glycosylated. So they looked at, so for example, SARS-1, SARS-CoV-2, and MERS, which are the viruses that cause either the epidemics or pandemics in humans. They are not that majorly glycosylated. They have about 22 to 23 glycosylation sites per spike protein monomer. Monomer meaning one spike protein, you need 3 of them to form the actual spike protein that is found on the surface of the virus. So that's not that many. They suspect that maybe this has to do with the fact that this allowed the virus to jump species, because the more glycosylation sites there might be, the more difficult it would be for the virus then to cross species, okay? And then if you look at some of the common cold viruses, which have been around for a very long time with us, then they have a lot more glycosylation sites. They can have somewhere between 26 to 31 glycosylation sites. And some of these viruses have been with us for a long, long time. And this is really neat. They basically, because of that, some of these viruses we've studied for a very, very long time, including how the glycosylation sites have changed over time in these viruses. So for example, they looked at influenza A virus, and what they mentioned is that for influenza A virus, the new glycosylation site was introduced approximately somewhere between every 5 to 7 years, okay? And when it comes to one of the common cold viruses, I think it was OC43, I think, then that one has been with us, or at least we've been aware of it for at least 60 years now. And for that one, it has a new glycosylation site every decade or so. And they mapped it to have this really cool graph showing how the glycosylation site increases over time. Now, there's one more common cold virus, I think it was HKU1, and that one has been around since just early 2000s, and that trend of increase is not as obvious as some of the other viruses. But basically what it means is that possibly virus might need to have long exposure time to humans in order for the glycosylation sites to be appropriately adopted by the virus. And perhaps this, while obviously SARS-CoV-2 virus is mutating at an incredible rate, this might help to explain potentially as to why Dr. Bossche, who always thinks that this evolution will happen in mere weeks, why he might potentially be wrong on this, and that evolution of introducing this new glycosylation site might just take a little bit longer than he anticipated, because it's clearly historically in these viruses, the introduction of new glycosylation sites take very, very long time. So maybe that could explain why he's off with his timing. But nevertheless, look, BA.2.86 introduced not 1 but 2 glycosylation sites right at the same time. So who knows? But here's his theory. Here's a paper that almost completely supports his theory, right? This is very much in support of his theory. Not completely, but, and I'm not gonna get into how it doesn't maybe support his theory, but clearly what he's worried about, this paper demonstrates that it's indeed happening in that direction as yes, he suspects what might happen. All right, so but again, if you think that he might be right, and I don't think he will be, I'm gonna argue against him, then and you are mRNA vaccinated, then you can attempt to protect yourself with appropriate antivirals and go from there. And which I think is totally fine to do. There's nothing wrong with getting some antivirals on hand for yourself. You know, cost almost nothing, and I would also agree that it would be very wise to also practice very healthy living right now in order to make sure that you are boosting the quality of your immune system to the best quality. All right, one of the longest videos I've ever made. This is a single paper, but again, this was such a complex story. Hey, I can't make it any easier. I could have cut out some of the things maybe, but this is such a complex story, and absolutely mind-boggling story. I wanted to share you this entire paper and its complexity, and how Dr. Bossche is getting another goal in the battle between our scientific minds. All right, that's all I have for you right now, and thanks for your support. Please give us a like. Please subscribe to the channel if you haven't already. Please share the video, and also check out our Patreon account for additional information that do not make it to the YouTube channel. Please leave your comments. I check some of them occasionally as well, more frequently on Patreon, because it's just overwhelming. I will also mention really quickly, I'm sorry that I have been not responding to the cancer one. I get too overwhelmed with that one. There's too much sadness for me to be able to deal with all of it. That's the reason why, but I still check a little bit here and there, and I appreciate your support, and I look forward to you in the next installment. Bye, everyone.